Hello everybody. Today we are going to cover the male genitalia. So first, let's start with the testes. The two testes are located in the scrotum, which is a skin pouch outside from the abdominal cavity. Because of the lower temperature, that's the best for the spermiogenesis. So you know the gross anatomy of the testes, you know the exact counterparts of the abdominal wall layers forming the testicle capsules surrounding them. But underneath the capsular system, we have a dense, irregular connective tissue that called tunica albuginea. So the tunica albuginea surrounds tightly the testes. On this drawing, we can beautifully see the exact cavity of the tunica vaginalis forming the visceral and parietal laminae of them. So this is the tunica vaginalis, and this dense irregular connective tissue that called tunica albuginea, it's written here, divides the testis by its septa into small units. So call these units testicle lobules. These are the lobules, and each lobule contains 2 to 3 coiled tubular system tubules called seminiferous tubules. So these seminiferous tubules lined by germinal epithelium, where the exact maturation of the cells, the sperm cells, occur. So these kind of places are essential for the proper sperm formation. Then, closer to the medial side of the testis, these tubules become straight, called straight tubules, and the straight tubules at the medial side form a network called rete. This is the rete testis. Rete means network. You remember when we studied the rete elbow, rete genu as well. So that they were arterial networks. So here the straight tubules form a network called rete testis. And this medial side of the testis called mediastinum of the testis. So in the mediastinum of the testis, we can see a network, that's the rete testis. And from the rete, from the mediastinum, we have connecting structures, efferent tubules toward the epididymis, which is eventually a kind of C-shaped structure surrounding the testis. So between the head of the epididymis and testis, we have the efferent ductals or efferent tubules just conveying the sperm cells for the storage in the epididymis. There is another thing. I want to show the schematic drawing about the cellular types in the seminiferous tubules. So in the tubules, we have different types of cells, differently matured cell types. This is the exact wall of the seminiferous tubule. Eventually, it's a really compressed, multilaminated basal membrane, and really close to the membrane, or we can say they are resting on this membrane. These are the less developed cell types inside called spermatogonia. These are the spermatogonia, and they differentiate into primary spermatocytes, and the primary spermatocytes later become secondary spermatocytes. But on a normal routine histological slide, we cannot see the secondary spermatocytes because their lifetime is just a few hours. So eventually we can see them. Then they differentiate into spermatids, and the spermatids form the spermatozoa or also called sperm cells. So that the proper name of them is spermatozoa. Among the maturing cells, we have supportive cells called Sertoli cells. They eventually maintain a special microenvironment in the seminiferous tubules. Eventually, they monitorize the hormone level inside and separate the maturing cells. So let's see the slide. That's the 78. Yeah, here it is. This is a longitudinal section about the testis and epididymis. Here we have the testis. That's the epididymis. This is the head. That's the body of the epididymis. And just when we zoom in, here we can see the appendix testis. You remember this is the developmental remnant of the Müllerian duct. And then here we can see the exact cavity of the tunica vaginalis. So here, just looking at the two sides of that. So here we can see the mesothelial cells forming the lamina parietalis, and the lamina visceralis is on the surface of the tunica albuginea. So this is it. That's the dense irregular connective tissue, and this capsule divides the testis into lobules. There is another thing, we cannot see the medial side on this section. I mean the rete testis and neither the mediastinum, but what we can see, the tubules, so these tubules, the small lumens, indicate the eventually the efferent tubules. So these are the connection between the testis and epididymis, the epididymic tubules. So the efferent tubules are there, and when we just zoom in, by the way, here we can see the septa. So these are the septa, followed by vessels, definitely. Also, so the dense irregular connective tissue forms septa. Now let's see a tubule, a seminiferous tubule. Which one is the best? Maybe it's better. Now look at that. This is a seminiferous tubule here, there, and at the bottom we can find spermatogonia. So these darkly stained, dark, small round-shaped structures. This is a spermatogonium. This is a spermatogonium. This is another one. And they differentiate into primary spermatocytes. This is it. That is another one. And these are also. And these cells here, with the prominent nucleoli, these are the Sertoli cells, just separating these cells. And over them, here we can find spermatids. So these are the eventually the spermatids. And in the lumen, the conical elongated structures indicate the head of the sperm cells, also called spermatozoa. I show another one. Maybe it's a little bit better. So look at that. Here we have the darkly stained spermatogonia, these primary spermatocytes. there is a cell with prominent nucleolus. This is a Sertoli cell. That is another spermatogonia, primary spermatocytes, primary. Here we can see many cell types in the lumen. Now let's see them. So look at that again from the bottom. So spermatogonia, primary spermatocytes, that is also primary. There is a cell with a prominent nucleolus, that is another one. Moreover, we can see its huge cytoplasm. So approximately there. So this is a Sertoli cell. This is a Sertoli cell, and eventually between two Sertoli cells, we have a really well-developed junctional complex, mainly tight junctions. And the Sertoli cells, by the way, this is another one. The Sertoli cells separate the spermatogonia and early primary spermatocytes from the other developing cell types, the late primary spermatocytes, secondary spermatocytes, spermatogonia, and these are the spermatogonia, and spermatids, the smaller ones, these. And eventually call this kind of separation blood-testis barrier. This is the blood-testis barrier. That means the developing cells closer to the luminal side are in a special microenvironment, and the own proteins from the bloodstream cannot reach them. So eventually, this special microenvironment essential for the spermiogenesis and the tight junctional complexes of the Sertoli cells keep them up. So, okay, so primary spermatocytes here. Here we have these round-shaped structures. These are spermatids, and this is a typical shape of the spermatozoa when the spermatids lose their cytoplasm. And in the lumen, we can find the sperm cells, we can find the spermatozoa. So here, this is another spermatid. It's just because of the section profile, it's also presented there. So spermatid, spermatozoa. These are also spermatozoa. Sertoli cell. Here we have the prominent nucleus. This is another Sertoli. Spermatids, these small round-shaped spermatids. Primary spermatocyte, primary spermatocyte. Okay. And just among the seminiferous tubules, so these are the walls. Among the seminiferous tubules, we can find big cells with a round-shaped nucleus and ovoid cytoplasm. These are the Leydig cells. Leydig cells Leydig cell. So Leydig cells, they secrete the testosterone. They are also called interstitial cells because they are located between the seminiferous tubules. So they are the hormone-producing cells of the testis. We have two cell types in the testis, which are not part of the germinal epithelium, eventually, or not developing cells. These are the Sertoli cells. These Sertoli cells are eventually supporting cells because they keep up the blood testis barrier, forming a special microenvironment for the maturing cells in the lumen. And they separate the spermatogonia and some of the early spermatocytes, and this barrier keeps them at the bottom. And then in the interstitial area, we can find hormone-producing cells called Leydig cells. Okay. And then when we are just going toward the epididymis, we can see the efferent tubules. So here, efferent tubules, the lining epithelium is changing, but here we can see some spermatozoa and preservated sperm. Definitely no germinal epithelium in the efferent tubules. And when we are just looking at the proper cross-section, how nice, look at that, cross-section of one epididymic tubule, epididymic ductule. So this is it. In the epididymis, the sperm eventually just stored, and the final maturation also occurs there. But here we have the more or less matured spermatozoa. So here we can see their heads. Then looking at the lining epithelium, in the epididymis, we can find pseudostratify columnar, eventually a pseudostratify columnar epithelium having stereocilia. So these long apical processes are stereocilia. You remember the stereocilia eventually are not able to move, that are not able to show active movements. They are just moved due to the fluid flow. So eventually they are really long, approximately 100-120 micrometer long structures at the apical surface. And in the epididymic tubule, we have these elongated cell types here. So the pseudostratify, sometimes we can see a stratified columnar, but mainly pseudostratify columnar. So these cells called principal cells. These are the nuclei of the principal cells. And at the bottom, we can find the basal cells. That's it. And this is the basal lamina. So these are these tall columnar cells are the principal cells, and we have the basal cells. So again, let's back to this, the efferent tubules. Where we can find also columnar cells. Look at that. They're columnar cells. They are somewhere ciliated, somewhere non-ciliated. But definitely the epithelium, the lining epithelium, is much more thinner than in the epididymic tubules. Now that was the epididymis. Okay, let's move on. Let's see the spermatochord, which is number 90. Yeah. So spermatochord, you know, also represents some of the testicle capsules. So we have external spermatic fascia, cremasteric muscle and its fascia, and internal spermatic fascia. So when we are looking at this structure, which is a trichrome stained structure, therefore the collagen fibers are appearing blue, the red blood cells in orange, nuclei in red. So these blue color indicates the external spermatic, this is the external spermatic fascia. It's too thick because it belongs to the inguinal ligament. So this is the inguinal ligament, and here this is the external spermatic fascia, and underneath these red structure here, these are the fibers. These are the fibers of the cremasteric muscles surrounded by its epimysium. So here, there, this is a typical striated muscle. This is a tangential section of it, and I think, yeah, we can find some part of the here. So proper cross-section about the striated muscle fibers. So here we have, they are multinucleated. These are the nuclei, you remember, just underneath the sarcolemma, and this is the connective tissue coat surrounding it. So that was the middle layer, the cremasteric muscle and its fascia, and then maybe this coat. Eventually, this is the internal spermatic fascia here. Related to the muscle, we have the cremasteric vessels. So this is an artery, definitely. Red blood cells, full of red blood cells in the lumen, really prominent muscularis middle layer. And by the way, here we can see this line. Just look at that. This is the internal elastic lamina. That's the adventitia. And there is a vein. Okay, that's another one. So cremasteric vessels are also presented. Now let's see the content of the spermatic cord. The most important, the prominent structure in the spermatic cord is a really thick muscular tube. This is the vas deferens. This is the vas deferens, followed by vessels, the differential artery, differential veins. So these are. And in the different duct, we have a simple columnar, or somewhere pseudostratified, but it seems columnar. So simple columnar, maybe these nuclei there suggest the pseudostratified appearance. So the simple columnar or pseudostratified columnar lining epithelium with microvilli, long microvilli. So this something at the top are long microvilli. In the propria, we have here elastic fibers. So this is the propria of it. So just a few cellular elements, connective tissue, elements we have. And then just look at that. This area here, this is it. That's the muscularis externa. So this is a typical structure, like the gallbladder, like the in female genitalia. That's the fallopian tube and ureter, which show no muscularis mucosae and no submucosa. So in the vas deferens, we can see only the lamina propria here and the muscularis externa. So this is the muscularis externa. Many layers are presented. So there is an inner and outer longitudinal layer. This is the inner outer longitudinal, and there is a middle circular layer. So 3 prominent layers. You remember the vas deferens carries the sperm from the epididymis toward the abdominal cavity, toward the prostatic part of the urethra, so during the ejaculation. So it's full of muscle, showing frequent contractions, pushing the sperm cells toward the prostatic urethra. So this is the epithelium, simple columnar, somewhere pseudostratified columnar epithelium with microvilli. There is the lamina propria with elastic fibers. Here also can see some connective tissue fibers. Here we have collagen fibers as well. And then no muscularis mucosae, no submucosa, just the 3 layers of the muscularis externa. This is it, inner and outer longitudinal and middle circular layer. And there is a loose connective tissue here. There is well presented connective tissue elements, vessels, nerves, and adipocytes, many capillaries. There are a thicker connective tissue structure, so it represents adventitia. Okay, in the spermatic cord, beside the vas deferens, we can find vessels. These are the main ones. So look at that. Look at the difference between these and this. So this is the artery. This is the artery. This is the testicular artery here. Endothelial cells forming the tunica intima. There is the subendothelial layer. By the way, here we can see the muscular. This is the internal elastic membrane. That's the one. So this is the intima. That one is the media. This is a nice muscular artery. And this is the adventitia. So that's the testicular artery. And you know, we have the pampiniform plexus, which eventually the tributaries of the gonadal veins, the testicular vein. So here we have the veins, so beautifully seen. The nice venous lumens. Here we can see the nuclei of the, that's the proper one, nuclei of the endothelial cells, subendothelial layer. There is a tunica media. That's the tunica media. Maybe that side is a little bit better. And this is the adventitia, which fuses with the environment. So many lumens here form the pampiniform plexus. Pampiniform plexus is a really well-developed venous network, just covering the components of the spermatic cord. Only one artery, that's the testicular artery. Okay, beside of that, we can find nerve components. So you don't need to know them. So, but these are a cross-section of peripheral nerves, because in the spermatic cord, we have the genital branch of genitofemoral nerve. You remember this nerve innervates the cremasteric muscle. And this nerve also presented at the surface of the spermatic cord. And sympathetic and parasympathetic nerves are also inside. Okay. Let's move on. Our next slide is the number. That's the prostate. That's the prostate. Yeah. 81. Here it is. So prostate is surrounded by a capsule. This is a capsule. This is a dense irregular connective tissue. Capsule here. That's the one. Beautifully seen. Fibrosards, coarse collagen bundles forming it. And you remember it has a really important, it has a really thick capsule because eventually it keeps the urinary bladder, holds the urinary bladder and fuses with its fascia as well, its capsule as well. So there is another importance of the prostatic capsule has, so the support of the bladder. And when we are just looking at the small magnification of the prostate, we can see a big lumen. That's the prostatic urethra. That's the lumen of the prostatic urethra. And in the lumen, there is a projection called seminal caulicle. Seminal caulicle. This is the seminal caulicle. It's just a projection. This seminal caulicle represents two openings for the ejaculatory ducts. And there is only one opening for the prostatic utricle. Prostatic utricle. The ejaculatory ducts, you know them very well. So these openings eventually made by the union of the excretory duct of seminal vesicle, producing semen, and the vas deferens. So the two ducts unite, forming the ejaculatory ducts. Vas deferens carries sperm cells. Excretory duct of seminal vesicle carries semen. So, and they form the ejaculatory, which open into the prostatic urethra and release their juices during the ejaculation. Prostatic utricle, it's a blind pouch. It's just a developmental remnant of the Müllerian duct. So the prostatic utricle is a developmental remnant. It's a small pouch of the Müllerian duct. It has a funny name, so it's called the uterus of the male. So, but it's extremely small. We cannot see it on this specimen, on this slide. We can see only the seminal caulicle. Then, so really close to the luminar surface, we have glands, and far away from the prostatic urethra, we have also big lumens. So we have glands everywhere in the prostate because it's we can talk about two zones. So we have internal zone and peripheral zone. So the peripheral zone represents the huge lumens there. So that's the periphery. The huge lumens indicating the main glands. So these are the main glands. And then the internal zone represents the mucosal and submucosal glands. In the main glands, in the main glands, we have a simple cuboidal lining epithelium. So there, that's cuboidal. Here, somewhere columnar, it's cuboidal. So also it's tall cuboidal. So we have these simple lining epithelium. So these are the main glands at the periphery. And there is another thing. In the lumens, we can find these kind of precipitated materials. These are, look firm, preservated some things called amyloid bodies. So amyloid bodies also called corpora amylacea. Corpora amylacea, so amyloid bodies, these are eventually the degradative materials of the glands, and because they are just presented because of the aging, causing the exact firm touch of the prostate during the manual examination. So we have smaller, bigger prostatic or amyloid bodies. Here, look at that. It's a concentrically preservated material in the glands, and closer to the lumen, we have glands there again. But these are different. They are in the internal zone, and the lining epithelium is called pseudostratified. So it's called pseudostratified lining epithelium. Just look at that. Here, there. So the many levels, the nuclei in many levels. So pseudostratified appearance also. And these are the submucosal glands, and really close to the surface. Also, we have mucosal glands showing also pseudostratified columnar cells lining these internal type of glands. So we have mucosal, submucosal, and main glands. Mucosal here, submucosal, and these are the main glands. And these glands embedded in a specific tissue called fibromuscular connective tissue. This is the fibromuscular connective tissue, beautiful aspect. Here we can see the ovoid, larger, elongated nuclei of the smooth muscle cells, and among them here we have, this is a typical of the fibrocyte, so we have connective tissue, mainly collagen fibers. So the mix of the smooth muscle and collagen fibers form this typical type of connective tissue, or the mix of connective tissue, which is really typical for the prostate, called fibromuscular connective tissue. Fibromuscular connective tissue, this is the typical environment for the main and submucosal and mucosal glands. So, yeah. So back to the small magnification. Prostate is surrounded by a dense irregular connective tissue there, and this capsule fused by the bladder. In the prostate gland. We can find at the periphery, the peripheral zone, main glands lined by simple columnar cuboidal cells. The lumens represents amyloid bodies. These are degradative materials inside. Then we have submucosal glands. They are also, can show here, concretions, this kind of amyloid bodies. So submucosal glands, mucosal glands, and also there is the lumen of the prostatic urethra. Into the lumen there is a projection called caulicle, seminal caulicle, having 2 openings for the ejaculatory ducts and 1 opening, a blind pouch, for the prostatic utricle, which is the developmental remnant of the Müllerian duct. And the connective tissue, the stroma, is fibromuscular. So fibromuscular connective tissue the prostate has. Okay. Our next is the seminal vesicle. That's the 89. Sorry, yeah, 89. This is it, 89. It's a cubular organ located at the posterior inferior side of the bladder. You know, this is responsible for the semen production beside the prostate, and the seminal vesicle secretes the majority, approximately the 70% of the semen, and also secretes fructose in the semen, providing extra energy for the sperm cells. So it's a coiled cubular glands showing these kind of irregular lumens. When we are just look at the surface, the internal surface, we can see the lining epithelium, which is simple cuboidal epithelium. So this is it. That's the one. So simple cuboidal or somewhere stratified cuboidal because of the appearance. And these brownish granules are really typical inside of the cells. Lamina propria is a cellular rich connective tissue. So simple cuboidal or pseudostratified appearance of the cells, cellular rich lamina propria. And again, I just zoom out and look at that. There is something weird. There's no muscularis mucosae and no submucosa. Just immediately underneath the propria, we have the muscularis externa. In the muscularis externa, we have inner circular outer longitudinal layers, but I'm trying to find a nice aspect, a nice view. So here, look at that, inner circular and outer longitudinal. Inner circular, outer longitudinal layer. Extremely thick muscle layers, again here. Inner circular, outer longitudinal muscle layers. And there is a connective tissue covering, that is adventitia, because, you know, it's infraperitoneal structure because it's located inferior at the inferior portion of the bladder. So when we're talking about the seminal vesicle, this is a highly coiled tubular gland secreting semen and fructose. In the lumen, we can find simple cuboidal, that's cuboidal. So here we can see these folds there. the presence of the red blood cells here is definitely artificial. So simple columnar cuboidal cells, the lamina propria is cellular rich here, and underneath it represents neither muscularis mucosae nor submucosa. Immediately muscularis externa begins there. So just looking at this aspect, inner circular, outer longitudinal fibers are presented, and the outermost layer is loose connective tissue here, there, because it's infraperitoneal organ, so definitely no attachment with the serosa. Okay. Our last slide is the penis. That's number 7. Yeah, that's the one. Yeah. This is a Verhoef staining showing the fibers, the connective tissue fibers. Here beautifully see the two erectile bodies called corpora cavernosa, so the corpora cavernosa and corpus spongiosum here. And the surface, just looking at the surface, it's a typical stratified keratinized squamous epithelium. So I'm just looking at this. Obviously it's a thin skin, but there is an important stuff. So these brownish somethings here, these are melanocytes because it's a pigmented skin. So these are pigment cells or melanocytes among the stratum basale cells. And then here we have the stratum polygonale or spinosum. We have granulosum, there is the granulosum, but no lucidum, and there is a thin keratin layer. So, look at that. Now it's much more better. So the melanin pigments are beautifully seen in the melanocytes, just among the stratum basale cells, these brownish substances. Yeah, again, stratum basale, stratum spinosum, granulosum, no lucidum, just corneum here. Okay, then underneath these connective tissue fibers are well presented. So the look at that. The Verhoef stains the connective tissue elements into the brown and dark colors. And underneath the skin, so underneath the skin, we have the tunica dartos. We have the tunica dartos, which is eventually just a subcutaneous tissue here. That's the tunica dartos. There. You remember when we studied the capsules of the testis, we mentioned related to the tunica dartos is just a few connective tissue elements and smooth muscle fibers. So look at that. Here we have these connective tissue cells and smooth muscle just among them. So this is the tunica dartos. And then this, it looks like multilamellated or something like that. So the well-packed, more dense area is also presented or layered. This is the penile fascia. This is the penile fascia. There. That's the penile fascia. Okay. Now let's see the covering of the corpora. So this is also called tunica albuginea. So this is also called tunica albuginea. This is a dense irregular connective tissue there. And corpora cavernosa, so this, and corpus spongiosum, also covered by tunica albuginea, but it's much more thinner than around the corpora cavernosa. Then, just outside from the corpora cavernosa, onto the surface of it, we can find some vessels, some lumens of the vessels. So at the superior to the corpora cavernosa, on the dorsal surface of the penis, we can find the lumens of the dorsal vein, the dorsal penile vein. So these lumens are belonging to the dorsal penile veins lumens. These are. And then at the lateral side of the corpus cavernosus, we have the dorsal artery. So look at that, really nice. This is the tunica intima. Here we have the internal elastic membrane, tunica media, and there is the adventitia. So that is the artery. The arteries are presented on the both sides of the corpora cavernosa. But I'm not sure. Maybe it's a branch of it, so it's not a proper lumen of the artery. But that side, that is much more better. So that's better. So this is an artery. So the arteries are located at the both sides of the corpora cavernosa, just underneath the penile fascia. And on the dorsal surface, we have the lumens of one vein, the dorsal penile vein, but the many subbranches of them are presented. Okay, then inside the corpora cavernosa, we have sinuses, vascular caves, just they able to receive the blood during the erection, causing the exact erected penis. And inside, almost in the middle, we can find the deep arteries, the lumens of the deep arteries. I think just only one lumen is visible here. So maybe it's a sub-branch of it, of the deep artery. That's the one. And somewhere there, but I think we cannot see. Yeah, we can see another one on the other side. So it's not on the section profile, just only this. So the two sides of the two corpora cavernosa represents these deep arteries. Again, I just want to summarize the vessels. So the dorsal penile artery, that's the vein. That's the vein, dorsal penile vein. That's the dorsal artery at the both sides, and inside we have the deep artery. Okay, at the both sides, but not on this side. Just there. The other is not presented because of the section profile. So in the corpora cavernosa, we can find sinuses, vascular sinuses. They receive the blood, so we can see eventually just a connective tissue mass forming the wall of the sinuses. But the lumens for this here, so these greenish black structures indicate the connective tissue fibers inside, and also there is a lumen. Red blood cells are also presented. So here. Okay, so vascular sinuses are in the corpora cavernosa. And these are connective tissue septa, just from the tunica albuginea, dividing the corpora cavernosa. Now let's see the corpus spongiosum, having the lumen of the urethra. This is the spongy part of the male urethra, and then it's also surrounded by tunica albuginea, but it's much more thinner. So dense irregular connective tissue surrounds it, and also in the corpus spongiosum we have numerous vessels just supplying the spongy part of the urethra, and there is an important structure inside in the luminal walls. These protrusions of the wall are presented everywhere, called Ebner's cushions. So this. So Ebner's cushion, these are all Ebner's cushions. They are also presented in the corpora cavernosa, but because of the really compressed numerous blood sinuses, we cannot see them beautifully. Therefore, we usually can demonstrate them in the corpus spongiosum. So Ebner's cushion. I'm going to show more. So for example, that's the lumen. That's the lumen of the vessel, and this is another cushion. That is another lumen. So here we have another cushion. Here, and there is the Ebner's cushion. These are also big cushions here, there. You know, during eventually the erection, when after the blood inflow and the intra cavernous pressure is getting higher, because the veins at the surface compressed, and only the deep veins can eventually convey the deoxygenated blood, convey the blood toward the body. Therefore, there is a limited vascular drainage. So because of the Ebner's cushions and because of the superficial veins. So then when we are just looking at the surface of the urethra, we can see the stratified columnar lining epithelium. So that is a stratified columnar lining epithelium. Maybe it's better aspect. So this is it. Here we can see the basement membrane there. And related to the spongy urethra, we have some glands. Oh, look at that. What a big Ebner's cushion here. No, it's not that bad because this area represents these glands. These are the Littray's glands, the juice lubricates the spongy part of the urethra before the ejaculation. So these glands here, Littrase glands, or paraurethral, also called paraurethral glands, they eventually lubricates the spongy part. But now we cannot see the exact exit. So this is a long tubular gland there toward the spongy urethra. These are also, yes, these. So this kind of mucus secreting glands are the Littrase glands here. They're really close, but we cannot see their exit, the excretory duct toward the spongy part. Okay. So that was the penis. Okay, our next time we are gonna cover the female genitalia. Okay, bye-bye.